So now let us see about the silent features of this double helical structure of DNA. Here we see the single chain of the DNA. So this is the 5' to 3' and here will another, here this chain will pair with the another chain and it will make double helical structure. So here adenine pair with the thymine, it is present here. Then anti-parallel strand of DNA. Here phosphate and sugar. Here nitrogen base, and then it is so this is 3' and here this is the 5' end. It makes the double stranded DNA. As you can see, this is the first chain and this is the second chain, and both the chain are going anti-parallel to each other. Okay, so now let us discuss about silent features of DNA. These silent features are proposed by the James Watson and Francis Crick by the process called, they found that by the process called X-ray diffraction. Now let us see what they proposed in the double helical structure of DNA. They said that DNA made up of two polynucleotide chain. Okay, so this is single chain of DNA and this is the another chain of DNA. Watson and Crick said that DNA is made up of two polynucleotide chain. Here this is the polynucleotide chain which is made up of phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay. The chain is anti-parallel to each other, as we have seen here that one chain go through the 5' to 3' end and another go to the 3' to 5' end. Okay. Now the end base pair for the hydrogen bond means here this base pair adenine and thymine is paired by the bond called hydrogen bond. This can be asked in exam. Hydrogen bond. Two base pairs are present or linked by the hydrogen bond in the DNA. Here it is a nitrogen base and hydrogen bond present between two nitrogen base pair. Two coil right-handedly fashion means it is a right-handedly fashion. Okay. Now two coil. Now the another silent feature is this DNA have 10 base pair in each turn. Okay, and the length of this turn, complete turn, one turn, is the length of complete one turn is 3.4 nanometer, and the distance between two base pair or two nucleotides is 0.34 nanometer. So it is a distance between two nucleotides. The distance between two nucleotides is 0.34 nanometer, where the one turn, the length of one turn is 3.4 nanometer, and in one turn there is 10 nucleotides present. Okay. So this is about the structure of DNA. Here I would like to mention that in these base pairs between adenine and thymine, there is 2 hydrogen bond, and in guanine and cytosine it is linked by the 3 hydrogen bond. Okay, so here if here nitrogen base is cytosine, then on other strand the nitrogen base will be guanine, and they both are linked by 3 hydrogen bond. Okay, so this is about structure of DNA. Now Watson and Crick also suggested that this DNA makes copies of themselves. This DNA makes copies of themselves. Okay, when it is in a synthetic phase, it will make 2 copies of themselves. So this process known as replication, it is known as DNA replication. So what is DNA replication? DNA replication means the process by which DNA makes copies of themselves, known as DNA replication. Watson and Crick also suggested, Francis Crick suggested that the how DNA expressed in a different cell, that first of all by the process called central dogma of molecular biology. So what is this central dogma of molecular biology? They said DNA, here it is DNA. How they express? What is central dogma of molecular biology? Central dogma of molecular biology says that how DNA is a synthesis or express themselves in a cell or organism. So look at here how they express. First of all, DNA makes copies of themselves. Here we see DNA makes copies of themselves, known as DNA replication. Okay. Then later DNA makes RNA. This process known as transcription. And then later RNA makes protein. So this is known as translation. From central dogma of molecular biology, one MCQ must become that what is replication? If DNA makes copies of themselves, known as DNA replication. If DNA makes RNA, the process known as transcription. And if RNA makes protein, this process known as translation. These processes, we will going to discuss this process in detail in later classes. Now in next class we will discuss about how this DNA is packaged in the cell, because we know that in each cell there is 2 meter long DNA is present in a cell, and it is necessary to pack this DNA in such a manner that it require less space in a cell or in a nucleus. So in next class we will discuss about packaging of DNA, that how DNA is packaged in a particular manner in a nucleus. Okay, so thank you friends. Hello students. So in previous classes we are starting a new chapter number 6, the molecular basis of inheritance, in which we discuss about structure of DNA. We know that structure of DNA. This is double helical structure of DNA. We discuss about its ultrastructure and the components by which the DNA is made. We already discussed all these things. Now today we are going to discuss about how this DNA is packaged in a cell or nucleus. So let us start today's class. We know that the length of DNA, by how we can find out the length of DNA. We know that in this DNA, between each pair, each nucleotide, the distance between each nucleotide is 0.34 nanometer, and the number of nucleotides present in a human genome is, in human cell, is 3.3 multiplied by 10 power to 9, which means by multiplying this length with this number of nucleotides, we can get the actual length of the DNA in a cell. So by calculating this, we find that the actual DNA length is 2.2 meter, around 2.2 meter in a particular cell. 2 meter. Now you know the diameter of cell and diameter of nucleus. So it is necessary to package in such a manner that this DNA require a less amount or less space in the cell. Okay, so how this DNA is packaged in the cell, that we will discuss in today's class. Firstly, in prokaryotic, we know that prokaryotes are primitive organisms and they don't have nucleus, well developed nucleus. So this is a prokaryotic cell in which DNA is present in a naked, without nuclear membrane. So in this DNA, the DNA in prokaryotes is wrapped around the protein, positive charged protein known as histone protein. Okay, in a simple manner. Now let us discuss about packaging in eukaryotic cell. So let us discuss. We know that this is the structure of chromosome, which is found in nucleus or cell during reproductive or division phase. Okay, so let us zoom this chromosome. What we will get? By zooming this chromosome, we will able to see that DNA is, the double stranded DNA is wrapped around the protein called histone protein. That can be asked in exam, that in a cell DNA is packaging in the which kind of protein? So this is histone protein, and this histone protein is positively charged. Okay, now let us see. This histone protein is organized in such a manner that 8 molecules unite with each other and form nucleosome. So the histone protein is made up of 8 proteins, and these all are made up and make a histone octamer. So this is called histone octamer. And when this DNA is wrapped around this histone octamer, this structure known as nucleosome. This structure is known as nucleosome. Now what is nucleosome? When DNA is wrapped around the histone octamer known as nucleosome. Okay. Now in each nucleosome, this is nucleosome. Now in each nucleosome there is 200 base pair. Means the length of this DNA is 200 base pair. This from here to here. The length of this DNA is 200 base pair. And we know that during a resting stage or interphase, DNA loses the structure of chromosome and form a chromatin-like structure, which is network-like structure, which is known as chromatin. And this chromatin, what is this chromatin? Chromatin is the repeating unit of nucleosome. मीन्स क्रोमेटिन क्या है? तो क्रोमेटिन इस न्यूक्लियोसोम का रिपीटिंग, बार बार ये न्यूक्लियोसोम रिपीट होके एक क्रोमेटिन बनाएगा. ओके. नाउ इन क्रोमेटिन देयर इज 2 रीजन. फर्स्ट वन इज लूजली पैक्ड रीजन, which is when we stain that particular region, it will look like lightly stained, and this region known as euchromatin. And the another region which is darkly stained and densely packed in cell known as heterochromatin. Okay. And the euchromatin is ट्रांसक्रिप्शनली एक्टिव क्रोमेटिन. मीन्स वो ट्रांसक्रिप्शनली एक्टिव होगा जिसमें से रिप्लिकेशन और ट्रांसक्रिप्शन एक्टिव होगी प्रोसेस. वेयर इन हेटेरोक्रोमेटिन इट इज इनएक्टिव. मीन्स द रीजन वेयर हेटेरोक्रोमेटिन इज प्रेजेंट विच इज ट्रांसक्रिप्शनली इनएक्टिव. नाउ द नेक्स्ट पॉइंट इज search for genetic material, which means during early time we know that something is passing from parents to offspring, and some factor which is present, as Mendel said that some factors are present in the human cell or nucleus which is responsible for the transfer of particular trait or particular character. So there people that time very interested to know that which kind of structure is responsible for the transfer of this trait from parents to their offspring or one generation to the another generation. That time people think that the structure of protein is very complicated, so they think that protein is responsible for the transfer of trait or character from the one generation to the another generation. But later by experiments they found that actually protein is not responsible for this transfer of character. The DNA is responsible for this. So how this man conclude that DNA is responsible for the transfer of trait? They conclude by the certain experiment which is conducted by different scientists. So let us see this kind of experiments. First experiment was conducted by Griffith in 1928. The name of that experiment is transforming principle. Means what is that? A particular thing which transfer from one offspring to another offspring, one organism to another organism. So what Griffith does in experiment, he take one bacteria. The name of that bacteria is Streptococcus pneumoniae, which is responsible for the production of pneumonia in an animal. So this Griffith take 2 different strain. First strain is S strain known as smooth strain, and another is a R strain, means rough strain. In smooth strain a capsule or polysaccharide layer is found on the colony of this bacteria. Means if this is bacteria, in smooth strain, one more layer present is around the bacteria, which give them more resistance. Where in R strain, this kind of bacteria don't have this shiny layer around them, so they are not resistant to the particular chemicals which is or enzyme which is produced in our animal's body. So what will happen here? Smooth strain bacteria is not, it will not die in animal body, so and it will produce a disease known as pneumonia. So this strain is known as virulent strain. And this rough strain known as avirulent strain. Means this strain is not producing pneumonia in animal. Now what he does, we know that he take two different strains of the bacteria and introduce this bacteria in mice. What will happen? First he introduce smooth strain bacteria having a shiny layer around them into mice. We know that this bacteria is virulent, so what will happen? The mice will die. Okay, we know that. Now he do another thing, that he take a rough bacteria and introduce into the mice. Then what is the result of this? The mice will live. Means mice zinda रहेंगे. Okay. Now this is, we know that rough strain is not producing pneumonia, so the mice is live. Okay. Now the another thing what he does, important thing is, he does a smooth strain bacteria, take smooth strain bacteria and heat them to know whether by heating this bacteria stay alive or they will die. After heating, they introduce this bacteria into mice. As a result of this mice will live. Means mice जिंदा रह गई. Or in last step, what he does, the important step where he take the rough strain bacteria which is alive, where smooth strain bacteria after heating, we know that after heating this bacteria may die, but after mixing of this, they mix both this strain and introduce into the mice. As a result of this, surprisingly he get mice dying. Now they think that I introduce a rough strain bacteria, but somehow this bacteria convert into smooth strain bacteria, which by the smooth strain die bacteria, which means something is from S strain bacteria is passing to the R rough strain bacteria. So he conclude that something is passing from this rough strain bacteria to smooth strain bacteria. Now what we understand from this Griffith experiment, we understand that something is passing from one organism to another organism in this case. Now we don't know that what kind of this structure, what kind of this molecule is transforming from a rough strain to smooth strain. Now another scientist come after Griffith. 3 more scientists come together and conducted one experiment. The name of the scientist is Avery, McLeod and McCarthy. And the name of this experiment is biochemical characterization of this factor or this DNA, RNA or protein. What he do? They take Histone bacteria, which is responsible for pneumonia. What they does? They purified three things from the three things from the Histone bacteria. First one is DNA, another one is RNA and protein. Now we can understand that what will they does or what will they do with this purified material or purified molecules. Now they introduce these molecules into rough stain mice by one. So they at the result they found that when they introduce protein in rough stain bacteria, they found that rough stain bacteria not converted into smooth stain bacteria. When they introduce RNA into the rough stain bacteria, they found that this bacteria also not transferred from rough stain to smooth stain. But when they introduce DNA into rough stain bacteria, DNA plus rough stain bacteria, they found that this rough stain bacteria is transferred to the smooth stain bacteria, which means here DNA is responsible for the transforming of rough stain bacteria to smooth stain bacteria. Now after this experiment of Avery, McLeod and McCarthy, people or they conclude that DNA is responsible for transforming of character or transforming of this rough stain bacteria into smooth stain bacteria.